Hello students, welcome to the EPG Pathshala. I am Dr. Snehata Panwar from the School of Life Sciences, Jawaharlal Nehru University, New Delhi. Today I am going to talk about the module Lipids in general with a specific focus on the structure and function of fatty acids from the paper Biomolecules and their interactions. The learning objective of this particular module is basically to introduce you all to the basic structure, nomenclature, and functions of lipids, and to understand as to how the hydrocarbon chain length of fatty acids can affect the properties of lipids. So what are lipids basically? Lipids, just like sugars, proteins, nucleic acid, are a class of biomolecules, but they differ from these other class of biomolecules in the sense that they do not form biological polymers. So these lipids have very low solubility in water but have a very high solubility in organic solvents like ether, acetone, benzene, chloroform, etc. So one unifying property of lipids is that they have low solubility in water, which means that they are water hating, or in other terms you can call them hydrophobic molecules. Fats and oils serve as storage forms of energy in living organisms and are basically derivatives of fatty acids. So, what are the properties of fatty acids that make them hydrophobic or water hating? So, lipids or fatty acids are largely hydrocarbon derivatives. Thus, they represent highly reduced forms of carbon, and their oxidation yields high amounts of energy. Lipids are required for the following important functions. They are important constituents of the lipid bilayer in all biological membranes. As you all know that the lipid bilayer is largely hydrophobic and is made up of phospholipids, cholesterol, sphingolipids, etc. So these lipids serve a very important function to maintain the structural integrity of all the biological membranes. Besides that, lipids also serve as energy stores. They also function as signaling molecules during intra or intercellular signaling events. I have already mentioned about lipids in general. Now let's have a look at the way lipids are classified. So there are different classes of lipids such as fatty acids, triacylglycerols, glycerophospholipids, which are also known as phospholipids, sphingolipids, steroids such as cholesterol, and there's another class of lipids that is classified as others, which includes components like waxes, terpenes, and eicosanoids. Let's look at what fatty acids are. Fatty acids are carboxylic acids with long chain hydrocarbon chains. Fatty acids do not exist in free form, and one exception to this is arachidonic acid. So how do these fatty acids usually exist in the cell? They usually exist in esterified forms as major components of various lipids. And these major components to which these fatty acids are complexed with can be triacylglycerols. In short, they're called as TAGs. The fatty acids that are linked to the glycerol backbone in the triacylglycerol have certain characteristics. One of the characteristics is that these fatty acids normally have 16 or 18 carbon residues and mostly will have even number of carbon atoms, usually between 14 to 24. Fatty acids are synthesized by the concatenation of C2 units, which is the converse of beta oxidation of fatty acids. When the fatty acids are broken down, they're broken down in C2 units. So further, the fatty acids are classified as either saturated fatty acids or as unsaturated fatty acids, When I say saturated fatty acid, I mean a fatty acid which has carbon-carbon single bonds. And when I say unsaturated fatty acid, I mean a fatty acid which has one or more double bond. Now, based on the number of double bonds, the unsaturated fatty acids can further be classified as monounsaturated. And as the name suggests, this unsaturated fatty acid would have one double bond, or they can be classified as polyunsaturated, which means that this unsaturated fatty acid can have more than one double bond. Let's have a look at how fatty acids are named and how are they depicted structurally. So the simplest way by which you can depict a fatty acid is by looking at their chemical formula, where you can see the number of carbon atoms that a particular fatty acid has, and you can see that this particular chemical formula shows that there is a double bond that is present in this fatty acid. So based on the number of carbon atoms that this particular fatty acid will have, this fatty acid is called as octadecanoic acid, which means that it has 18 carbon residues. The 1 here represents the presence of a single double bond. The delta 9 here shows that this single double bond is present between carbon 9 and carbon 10. This is a way by which fatty acids are generally depicted. Each of this tip represents a carbon atom which is bonded to hydrogen atoms. The carboxyl end is from where you start numbering the carbon atoms in a fatty acid chain. So this carbon at the carboxyl end is 1, and the one that is next to it is then 2, 3, 4, so on and so forth. The carbon adjacent to the carboxyl carbon is called an alpha carbon. So as you see here in the structure, that this particular fatty acid has 18 carbon residues, and that's why it's called octadecanoic acid. The double bond is at the position C9, so it's called 9-octadecanoic acid. If you look at another fatty acid which has more than one double bonds, you see that it is depicted as 25 delta 5, 8, 11, 14, 17, which means that this fatty acid, which is an eicosapentaenoic acid, has 20 carbon residues as in its chain, 5 double bonds, and these 5 double bonds are placed at positions 5, 8, 11, 14, and 17. This fatty acid, the eicosapentaenoic acid, is also called as an omega-3 fatty acid. So where is this omega carbon basically? The farthest end of the fatty acid chain, which is the methyl group, is called as the omega carbon. So when you say that it's an omega-3 fatty acid, you're basically counting the position of the double bond from this omega carbon. So the omega carbon, and then you count from here, the omega carbon is carbon 1, and then you count 2 and 3, and there is a double bond at the C3 position from the methyl terminal, and therefore this particular fatty acid is called an omega-3 fatty acid. The position of the double bonds 5, 8, 11, etc. are placed or can be counted from the carboxyl end. So you see that the first double bond is placed between carbon 5 and 6 if you count from the carboxyl end as 1, 2, 3, 4, and 5, followed by another double bond which is present between carbon 8 and 9. So this is the general way in which fatty acids are named. Here you see different fatty acids listed in the table on the left side, and on the right side, just by their names, you see the way they are structurally depicted. So stearic acid is one of the most common saturated fatty acids that you will come across in the textbooks. And as you look at the structure, you see that there is no double bond in this structure, which means that it is a saturated fatty acid, as compared to oleic acid, which has two double bonds and so is called an unsaturated fatty acid, followed by linoleic acid, which has three double bonds, and linolenic acid, which also has three double bonds. Both linoleic and linolenic acid are also called as omega fatty acids. Let's come back to the characteristics of fatty acids. I already mentioned that fatty acids cannot exist in free form. An exception to that is arachidonic acid. They usually exist in complex form, and they often exist in complex with triacylglycerols, or tags in short as they are called. So let's have a look at how fatty acids are complexed with a glycerol molecule. So glycerol has three hydroxyl groups, while the fatty acids have carboxyl groups. So the carboxyl group gets linked to these hydroxyl groups of glycerol through an ester linkage by removal of a water molecule, thereby giving rise to a glycerol backbone, which is now in ester linkage with three fatty acids. And this molecule is referred to as a triglyceride or a triacylglycerol molecule. The fatty acids that are attached to the hydroxyl groups of glycerol can either be identical. If all these fatty acids are identical, the triglyceride is called as a simple tag. If these fatty acids are different from each other, the triglyceride is called as a mixed tag. Let's look at the characteristics of the fatty acids that are attached to the glycerol backbone in a triacylglycerol molecule. Like I said before, these fatty acids predominantly are 16 or 18 carbon residues long and mostly will have even number of carbon atoms, usually between 14 to 24. The fatty acids are synthesized by concatenation of C2 units. This table lists the fatty acids that are commonly found linked to the glycerol backbone. For example, lauric acid is one of the most common saturated fatty acid that you will come across in all the textbooks. Myristic acid, palmitic acid, arachidic acid, etc. These fatty acids differ from each other in the number of carbon residues that they have. And as you see, the melting point of these fatty acids increases as the chain length of the fatty acids increases. So in other words, lauric acid has the smallest hydrocarbon chain, and so its melting point is about 45 degrees Celsius. As opposed to arachidic acid in this table, which has the highest number of carbon residues in its hydrocarbon chain, and therefore its melting point increases. So the melting point increases from 45 to 76 as the chain length of the fatty acid increases. And this characteristic of the fatty acid also affects the characteristic of the triacylglycerol molecule. This table, on the other hand, lists fatty acids that are unsaturated, which means that these fatty acids have double bonds, single or more than single double bonds present within them. So as examples, palmitoleic acid has a single bond, linoleic acid has two double bonds, arachidonic acid has more than single double bond. So as you see that as the degree of unsaturation or as the number of double bonds increases in these fatty acids, the melting point of these fatty acids reduces. So palmitoleic acid has a single double bond. Its melting point is 0 degree, as compared to arachidonic acid, which is more than one double bonds, and its melting point has gone down to minus 49 degrees Celsius. Let's now look at how the fatty acids are classified based on the presence of a single or a double bond. So they are classified as saturated if they have no double bonds, and as unsaturated if they have one or more double bonds. Unsaturated fatty acids are further classified as monounsaturated if they have one double bond, and as polyunsaturated if they have more than one double bond. In the unsaturated fatty acids, a common pattern exists in the location of the double bonds, which means that the double bond is usually between carbons 9 and 10 if you count the carbon from the carboxyl end of monounsaturated fatty acids, while other double bonds are majorly at positions carbon 12 and carbon 15 in case of polyunsaturated fatty acids, where arachidonic acid is an exception. Another characteristic of the polyunsaturated fatty acids, which I will hereby refer to as PUFAs, is that the double bond occurs at every third carbon atom and is separated by a methylene group. Hence, they are not conjugated as shown here in CH double bond CH, CH double bond CH, where this structure does not seem to have a separator, which is a methylene group in case of PUFAs. Oleic acid is the most common unsaturated fatty acid, and it is depicted as 18,1,9, which means that oleic acid has 18 carbon residues in its hydrocarbon chain, has a single double bond that is placed between carbon 9 and 10. All the double bonds that are present in the unsaturated fatty acids are always in a cis configuration in all naturally occurring fatty acids. So, what are the advantages of a cis bond in unsaturated fatty acid? The cis bond causes a kink or a bend in the fatty acid chain. Because of this kink or bend, the unsaturated fatty acids do not pack as closely as the saturated fatty acids. Van der Waals radii prevent them from packing closely to each other. So this loose packing, in fact, is making these unsaturated fatty acids more flexible and fluid. So all of these above features of the cis bond renders membranes, biological membranes or plasma membranes, more flexible and fluid. Here we see the difference between cis versus trans fatty acids. So you see a cis oleic acid structure and you see a trans oleic acid structure. You see that there is a bend in the cis oleic acid structure, while the trans oleic acid tends to be more linear. And why does this happen? The trans oleic fatty acids are generated artificially. They provide more linearity to the fatty acid structure, because of which they stack very closely to each other and therefore have high melting points. As opposed to the unsaturated fatty acids, which have the double bonds in cis configuration, like I already mentioned before, because of the cis configuration, there is a bend in the fatty acid, which prevents them from packing closely to each other. Thereby, these cis fatty acids have lower melting points as compared to the trans fatty acid. So these cis fatty acids, because they have lower melting points, they can exist as liquid at room temperature. They are generally considered good for health if consumed in moderate quantities. And like I said before, all the naturally occurring fatty acids have double bonds in cis configuration. The trans fatty acids, like I said, have high melting points because they provide linearity, or they make the fatty acids more linear, because of which they can pack very close to each other. Just imagine a box of chalks tightly packed, tightly stacked against each other. That is how a trans fatty acid will be packed, and so they are solid at room temperatures as compared to the cis fatty acid. So these trans fatty acids are harmful for health and are linked to cardiac diseases. How are these trans fatty acids obtained? So basically, these trans fatty acids come from processed food such as hydrogenated oil. So if you bubble hydrogen gas into a mixture of cis fatty acid, what you end up getting is trans fatty acids in that particular mixture. So unsaturated fats are normally hydrogenated in order to increase their shelf lives because their stability increases if you hydrogenate them. In that process of hydrogenation, what happens is hydrogen is added to the fatty acid, and so unsaturated fatty acid keeps getting converted to a saturated fatty acid, but it does not completely become saturated fatty acid because the unsaturated fatty acid will have more than one double bond present in it. So not all the double bonds are become saturated or hydrogenated, but this particular fatty acid, because of the bubbling of hydrogen gas into it, has become partially hydrogenated. So you would see food packs that mention contains partially hydrogenated oils. So if you see a food pack with this particular term mentioned, partially hydrogenated oil, you should understand that this particular food item contains a lot of trans fatty acid, which are risk factors and are linked to cardiac diseases. So this slide basically shows the characteristics of omega fatty acids. Omega fatty acids are polyunsaturated fatty acids or PUFAs, wherein the position of the double bond is counted from the methyl terminal. This methyl terminal is also called as the omega carbon, and it's at the other end of the chain, while the carboxyl end is on one end of the chain. So the omega fatty acids are of two kinds, omega-3 fatty acids and omega-6 fatty acids, such as alpha-linolenic acid, alpha-linoleic acid, which are majorly found in plant oils. Then there is an eicosapentaenoic acid, which is abbreviated as EPA here, and docosahexaenoic acid, which is abbreviated as DHA. Both these fatty acids are also omega fatty acids and are found in marine oils. So marine algae and phytoplanktons are source of these omega fatty acids. These fatty acids are essential for metabolism because they cannot be synthesized by humans, hence they are also called as essential fatty acids. Humans obtain these omega fatty acids like alpha-linoleic acid, which is about 18 carbon residues long, from their diet. Alpha-linoleic acid in turn is then used as a precursor to synthesize longer omega fatty acids like EPA, which is 20 carbon residues long or more. Here we see the characteristics of saturated fatty acids. We've already seen that there are saturated fatty acids, there are unsaturated fatty acids, and we know now that there can be a monounsaturated fatty acid, there can be a polyunsaturated fatty acid, and we've already seen now that omega fatty acids are a kind of polyunsaturated fatty acids, which are also referred to as essential fatty acids. So saturated fatty acids are highly flexible molecules. Why are they flexible? They are flexible, they do not have double bonds, so they allow free rotation about the carbon-carbon single bonds. Because of this free rotation, these fatty acids can attain a wide range of conformations. The fully extended conformation of these fatty acids is thermodynamically favorable because there is least steric interference with the neighboring methylene groups. Because of this fully extended conformation, these fatty acids pack very tightly together to form a solid at room temperature, such as butter. The melting points of these fatty acids increase with their molecular mass. If you remember, we already saw the list of fatty acids, a table where we had listed fatty acids and shown how with an increase in the carbon residues, the melting point of the fatty acids increases. So you have to remember that saturated fatty acids are flexible, they pack tightly together, and so they form a solid at room temperature, and their melting point increases with an increase in their molecular mass or with an increase in the number of carbon residues in the hydrocarbon chain. As opposed to the saturated fatty acid, the cis double bonds that are present in the unsaturated fatty acid lend a 30-degree bend in the hydrocarbon chain. This bend then interferes with the packing of unsaturated fatty acids in space and causes reduced van der Waals interactions. So the packing of these unsaturated fatty acids is loose, hence their melting point decreases with the degree of unsaturation, such as vegetable oil, which we all know is good for our health because it does not block our arteries. The loose packing of the unsaturated fatty acid also contributes to lipid fluidity, which is an important property of all biological membranes. So let's have a look at how the melting point of fatty acids with the same number of carbon atoms but with different number of double bonds will vary. So here you see that stearic acid is a saturated fatty acid because it doesn't have a double bond and it has the highest melting point of 69.6 degrees Celsius. If you look at linolenic acid, which has 3 double bonds but the same number of carbon atom as in stearic acid, you see that the melting point has gone down to minus 17 degrees Celsius, which indicates that the lower the degree of saturation, the lower would be the melting point. In this table, you see that saturated fatty acids here that are listed differ in the number of carbon residues in their hydrocarbon chain. So lauric acid has 12 carbons as opposed to lignoceric acid, which has 24 carbons. And melting point of these saturated fatty acids, as I have mentioned before, increases with an increase in the chain length of the fatty acid, which is depicted here in this table. So if you look at the melting point of lauric acid, it's 44.2, and if you look at the melting point of lignoceric acid, it is 84.2 degrees Celsius, again indicating that melting point of saturated fatty acids will increase with an increase in the chain length. So let's see what fats and oils are. Fats and oils consist largely of mixture of triacyl glycerols. And as we have already defined what triacyl glycerols are, these triacyl glycerols are also referred to as fatty acid triesters of glycerol. So the question that will immediately come to your mind if you think about the fact that both fats and oils consist a mixture of triacyl glycerols is that why are fats solid while oils are liquid at room temperature? So why is there a difference between fats and oils? So I can give you a hint. The hint is that plant oils are richer in unsaturated fatty acids compared to animal fats. And as we have already discussed before, because the unsaturated fatty acids are very high in plant oils, it makes them liquid at room temperature, while the animal fats have more of saturated fatty acids, thereby making them solids at room temperature. So since we've already discussed about omega fatty acids and we already have termed these omega fatty acids as essential fatty acids, let us have a look at what these essential fatty acids are and how are they synthesized in our body. So essential fatty acids, as I said, cannot be synthesized inside the human body. Therefore, they need to be obtained from our diets, mostly from plant sources. An exception to this is arachidonic acid, which is a C20 compound not found in plants, can only be synthesized by humans, and the precursor for arachidonic acid is linoleic acid, which is an essential fatty acid again. So it means that linoleic acid, which is an essential fatty acid, needs to be obtained from the diet, which in turn serves as a precursor for the synthesis of another fatty acid called arachidonic acid, which will then serve as a precursor for the synthesis of eicosanoids, which are 20 carbon residue compound like prostaglandins, thromboxanes, and leukotrienes. So prostaglandins, thromboxanes, leukotrienes collectively are referred to as eicosanoids. Prostaglandins, as we know, are intracellular hormone-like molecules that are not transported through the bloodstream to other cells for function. Instead, they function locally very close to their site of production and are also called as paracrine hormones. Here you see a table which lists different unsaturated fatty acids, and the ones that are marked with the arrow are the ones that are called as essential fatty acids. As we have already discussed, linoleic acid is one of them, followed by alpha linolenic acid, gamma linolenic acid, and eicosapentaenoic acid. So all these fatty acids are unsaturated. They are actually polyunsaturated. The double bonds are in cis configuration, but they are essential fatty acids because they cannot be obtained, because they cannot be synthesized by the human body. Instead, they have to be obtained from the diet. Here you see how linolenic and linoleic acid are depicted. So this is the carboxyl end on the right hand side. The omega carbon is marked on the left hand side, and if you start counting from the omega carbon end, the omega carbon is carbon 1 followed by the tip, which is the second carbon, followed by the third carbon, and you see that the double bond is placed between the third and the fourth carbon when counted from the omega carbon end. Similarly, in linoleic acid, you see that the double bond is placed between carbon 6 and 7. Therefore, linolenic acid is also called as an omega-3 fatty acid, while linoleic acid is called as an omega-6 fatty acid. As we now know that these fatty acids like linolenic and linoleic acid are essential fatty acids, which means that they have to be obtained from diet. So what are the different functions that these fatty acids serve in the body? So linolenic acid, which is an omega-3 fatty acid, serves as a precursor for the synthesis of eicosapentaenoic acid, which in turn serves as a precursor for the synthesis of a subtype of prostaglandin, which is PGE3. Linoleic acid, on the other hand, serves as a precursor for the formation of gamma-linolenic acid, which in turn synthesizes arachidonic acid, which in turn serves as a precursor for the synthesis of another subtype of prostaglandin, which is called PGE2. And we've already seen as to how arachidonic acid serves as a precursor for the synthesis of prostaglandin, thromboxanes, and leukotrienes. So arachidonic acid serves as the most important precursor for the synthesis of eicosanoids. Arachidonic acid itself is a PUFA with 4 double bonds. It is stored as the C2 ester of phosphatidylinositol and other phospholipids, and it is released from this ester linkage by the action of phospholipase A2. So what are the characteristics of eicosanoids and why are they important? Eicosanoids act at very low concentrations in the body. They cause pain and fever. They regulate blood pressure, blood coagulation, and reproduction. And unlike hormones, they are not transported to other organs. Instead, they function locally near cells that generate them. Eicosanoids decompose within seconds, a reason why their effect is limited. Here we see the various steps that are required for the synthesis of eicosanoids like thromboxane, PGF2 alpha, which is a prostaglandin, a leukotriene, etc., from arachidonic acid. So we've already seen that arachidonic acid serves as the precursor for the synthesis of thromboxanes or leukotrienes or prostaglandins. The first step that converts arachidonic acid to PGH2 is catalyzed by an enzyme called PGH2 synthase. This enzyme is also called as cyclooxygenase. There are 3 isoforms of this enzyme
COX-1, COX-2, and COX-3. PGH2, which is synthesized by PGH2 synthase, is then further converted to a prostacyclin, a prostaglandin, and a thromboxane. This enzyme, PGH2 synthase, is used as a target by certain drugs. For example, aspirin. Aspirin inhibits PGH2 synthase, thereby blocking the conversion of arachidonic acid to PGH2, which in turn will block the production of all the other eicosanoids. So, enzymes involved in the synthesis of the C20 compounds, like the PGH2 synthase, are major drug targets. And like I said, aspirin targets this enzyme and blocks the conversion of arachidonic acid to eicosanoids. Aspirin is also called as non-steroidal anti-inflammatory drug, or NSAIDs, as abbreviated here. These NSAIDs inhibit the synthesis of prostaglandins from arachidonic acid. Besides aspirin, ibuprofen, acetaminophen are also classified as non-steroidal anti-inflammatory drugs. So what is the mechanism of action of NSAIDs? Aspirin is widely used as an analgesic. We all know about that. And we also know that a low dose of this drug is reported to reduce heart attacks and strokes. So aspirin basically inhibits the synthesis of prostaglandins by acetylating a specific serine residue of PGH2 synthase, the enzyme that converts arachidonic acid to PGH2. Ibuprofen and acetaminophen, on the other hand, non-covalently bind to this enzyme and blocks its active site. So the mechanism of action of these drugs is different from each other, although all of them target the same enzyme, which is PGH2 synthase. The disadvantage of aspirin and ibuprofen is that they are non-specific drugs and hence can have adverse side effects. Hence, another isoform of PGH2 synthase, which is called COX-2, is currently being targeted for developing new drugs. PGH2 synthase has two isoforms, COX-1 and COX-2. COX-1 is the target for aspirin and ibuprofen. The three-dimensional structure of these isoforms are very similar, but the COX-2 active site channel is approximately 20% larger in volume than COX-1 due to amino acid differences between them. Inhibitors that have been synthesized for COX-2 are collectively referred to as COXIBs, such as rofecoxib and celecoxib. These drugs have an advantage over aspirin and ibuprofen in the sense that they do not produce the side effects like aspirin. Acetaminophen binds poorly to both COX-1 and COX-2. Instead, it binds to a third COX isozyme called COX-3. So far we've seen how important fatty acids are, and we've also now looked at the different kind of fatty acid that exist in the cell. So there are saturated fatty acids, unsaturated fatty acids. There are polyunsaturated fatty acids. Omega fatty acids are PUFAs, and they are also called as essential fatty acids. And we have now seen why these essential fatty acids are required by our body and how the biosynthetic pathway of the essential fatty acid can be targeted in order to generate drugs like aspirin and ibuprofen.
So, what is the take-home lesson that you have obtained from the series of lecture in this particular module? The first take-home lesson is that fatty acid-wise, you become what you eat, which means that there is a good fat and there is a bad fat. If you take good fats in your diet, you're healthy, and if you thrive on bad fats, you become unhealthy. So it means that fats in general have a far-reaching effect on our health. For example, saturated fats are considered bad fats. Saturated fats like butter, margarine, mayonnaise, etc., are the most harmful to human health compared to the polyunsaturated fatty acids that are present in vegetable oils or cooking oils, etc. Amongst the polyunsaturated fatty acids, we have already seen that the omega fatty acids are referred to as essential fatty acids. There are two kinds of omega fatty acids, omega 3 and omega 6, and we know how linoleic acid is obtained from the diet, which serves as a precursor for arachidonic acid, which in turn serves as a precursor for eicosanoids. So these essential fatty acids are so called because they cannot be synthesized by humans, hence they're obtained from the diet. So simply put together, the main lesson that you take back home from this lecture is that fat-wise, you become what you eat. Thank you.